Hello everyone and welcome to the second installment of Digested News Live here on the Genovis YouTube channel. My name is Andreas, I'm a senior scientist at Genovis, and today we are talking about O-glycans, the challenges that arise in characterizing O-glycosylated biopharmaceuticals, and the unique tools that Genovis has to offer to rise to these challenges. During this presentation, you have the chance to ask questions using the YouTube chat function, and we'll see whether we can answer them at the end of the presentation. Before we jump in, a few words about Genovis as a company. We are a small biotech company located in Lund in the south of Sweden, where we manufacture reagents for the biopharmaceutical industry that we call smart enzymes. When producing and developing biologics, there's a lot of analysis involved. These are large molecules with a lot of inherent heterogeneity. They're produced in living cells. So from early development through process to final manufacturing, there is a lot of analysis that needs to be done, often by liquid chromatography and or mass spectrometry. And we see here a pronounced need for quality reagents for sample preparation to make defined digestion of these very complex molecules so to get the most out of your LC-MS analyses. And that's where Genovis and the Genovis smart enzymes come in. What are smart enzymes? There's quite a few of them by now. We sell, for example, IgG proteases. You might have heard of Fabricator, and if you haven't, I recommend you watch the last installment of Digested News Live, where my colleague Jonathan Sjögren explained how to use this enzyme and the kind of applications where this is used. We also have IgG glycosidases, general arginine-specific protease, conjugation technology for antibodies, and the topic of today's talk, enzymes to analyze O-glycans. What are O-glycans and why are they difficult to analyze? There are many different types of O-glycans, but the most common one and the one we are talking about today is called mucine type O-glycan. It entails the addition of N-acetyl-galactosamine to serine and threonine residues of proteins as they pass through the Golgi apparatus. They're very structurally diverse depending on how these N-acetyl-galactosamines are elongated. There are different core structures, up to 8 of them. The most common 4 are shown on the slide here. And they're highly relevant in many different contexts, in many different biological processes and pathologies. They're also relevant for the biopharmaceutical industry as Fc fusion proteins and some of the other new formats that arise nowadays are O-glycosylated. Luckily for us, in the context of biopharmaceuticals, the O-glycans are often smaller and less complicated, mostly being of the core 1 type that is marked here in orange. An example for such a O-glycosylated biopharmaceutical is etanercept, shown here on the slide, which is a fusion protein between the Fc domain of IgG1 and the TNF-alpha receptor. It's used as an anti-inflammatory drug and is one of the top-selling biopharmaceuticals worldwide today. It has this heavily O-glycosylated domain in the TNFR region that is very difficult to analyze. Why are they difficult to analyze? Largely because the analytical toolbox is so limited. There are only few enzymes specific for O-glycans. There's no PNGase F-type enzyme to release all the O-glycans. And chemical methods to release them are often laborious and prone to artifacts. So what's left is peptide mapping approaches, where one needs to appropriately digest these heavily O-glycosylated proteins to do LC-MS analysis. And what happens in such an experiment, often these O-glycans form clusters where they're very close to each other. So if we take this model protein here with many core 1 O-glycans, if you would try to digest with trypsin, you end up with something like this. Very large chunks of protein that are heavily O-glycosylated, many different O-glycans on the same peptide. Very difficult to analyze and get any site-specific information out of this using LC-MS. Using other more unspecific proteases then makes data analysis harder, so Genovis offers another option, and that is one of our smart enzymes called ODERATOR. It is a O-glycan specific protease, which means it will digest N-terminally, specifically N-terminally to O-glycosylated serine or threonine residues. It doesn't cut unglycosylated serine or threonine residues or next to N-glycans. It likes these core 1 O-glycans found on biopharmaceutical, the best substrate, and we recommend removing any potential sialic acids on these core 1 glycans using our sialidase mix Silex, so prior to operator treatment. So if we move back to our cartoon model protein, if you would digest something like this with operator, what you would get is this. A large panel of overlapping O-glycopeptides which you can identify by LC-MS and map out all the different sites. Having these overlaps giving an additional level of confidence that your identifications are actually correct. What does this look like in practice? Here is an example using this Fc fusion protein etanercept, which we digest using operator in Silexo to get peptides, reduce alkylate, clean up on graphite, and then analyze by Hillick MS-MS. What do you get from this? You get a chromatogram full of O-glycopeptides where you identify all these different overlapping O-glycopeptides and map out all the different sites on the protein. No enrichment is needed, and a lot of site-specific information can be obtained without advanced fragmentation techniques such as ETD. It doesn't stop with this type of qualitative identification of O-glycosylation sites. You can also do quantitative comparisons. You can compare O-glycosylation patterns of a biopharmaceutical for comparability of batch to batch, for example, or like we've done here, between originator and biosimilars. So we've done the same workflow using the originator of etanercept and 2 biosimilars. And as you can see here from the chromatogram, the middle biosimilar shows a different relative abundance in 3 peptides marked in gray. All of these 3 peptides come from the same region of the protein and point to an underglycosylation on a single site in this one biosimilar. So we have a very specific and very sensitive assay to profile O-glycosylation and compare this between different batches or different preparations of this protein. As biopharmaceuticals move from early development on to clinical testing or final manufacturing, there's a need for a lot of routine analyses and quality control. Routine analysis often means that assays are preferred that do not require high-end instrumentation or expertise knowledge, such is the case for the high-resolution mass spectrometry. So we tried whether we could get similar results in comparing etanercept originator and its biosimilars without the need of a high-resolution mass spectrometer, but replacing it with the UV detector of the LC system or a very simple single quadrupole mass spectrometer where we can monitor the specific peptides that have previously been shown to be different in the different biosimilars. And the short answer is yes, we can get very similar results. We find the same 3 peptides that are different, also detected by UV or by the single quad MS, showing that this assay can be moved from initial characterization using high-res also down to routine QC type assays where you're just profiling and see whether the profile matches what you've previously seen or not. For the next few slides, I would like to leave the field of biopharmaceuticals and show you what people in the field of glycoproteomics have done with operators. So that's mostly basic research, academic research type applications where they're trying to map out O-glycosylation sites in tissues and cells and these type of samples. This is a publication that came out 2 years ago from Johns Hopkins in the States. And they developed a workflow they call XOO, which is based on operator digestion to map O-glycosylation site. The samples are digested using trypsin, and then these tryptic peptides are immobilized via their N-termini covalently onto a solid support. O-glycopeptides can then be specifically released using operator digestion, and as operator digests N-terminally to the O-glycan, the O-glycosylated part of that tryptic peptide will be released from the beads. The rest, plus all the non-glycosylated or N-glycosylated peptides, will stay in the solid support and can be removed. That gives a very clean O-glycopeptide sample that allows for very in-depth analysis of the O-glycosylation in that specific sample. They've done this using different types of samples, such as kidney tissue or serum or cell lines, and mapped over 3,000 different sites. They were also able to quantitatively compare O-glycosylation patterns in healthy kidney tissue versus tumor tissue, and map out the aberrant glycosylation patterns that arise during cancer. A very interesting publication that I highly recommend you take a look. There's also 2 more publications which used very similar approaches, this time more on purified proteins for an in-depth analysis of purified proteins using stuff like Zika viral capsid in the case of the analytical chemistry paper from the FDA, or the other one, hot off the press, just about 2 weeks old, where they took a very in-depth look at human coagulation factor 5 and its N and O-glycosylation patterns, also using very similar operator-based approaches. O-glycans are not only difficult to analyze, their complexity and heterogeneity can also make analyses of other quality attributes or post-installation modifications very difficult. For example, they can make LC-based separation difficult, or something like intact mass analysis by mass spectrometry. So sometimes there's a need to remove O-glycans to get at the underlying protein and characterize its modifications and heterogeneities. For that, Genovis offers O-glycer, which is a O-glycosidase. It specifically removes core 1 O-glycans that are so often found on biopharmaceuticals, and it absolutely requires pretreatment with Silexo. So Silexo will remove the sialic acids, O-glycer will remove the core 1 O-glycans, and you will end up with a pristine unmodified protein that you can use for your analysis. To show an example here, again using etanercept as an example, as our model protein, where we separated the FC part from the TNFR region using fabricated digestion, and then removed N-glycans by PNJZF, O-glycans by Silexo and O-glycer, and then analyzed the resulting proteins using middle-level LCMS. And this is what you get if you just do the fabricated digestion, the one on the left. You will end up with very complex spectra. All the O and N glycoforms overlap and give isobaric structures that are very hard to resolve, sometimes even hard to deconvolute. If you're removing the N-glycans using PNJZF, you start seeing some composition of the O-glycans, but it's still very difficult to map all the different peaks and identify all the underlying modifications. When you then add Silexo and O-glycer, you're basically done with one single peak corresponding to the unmodified protein, whose mass you can determine with very high accuracy. It also becomes clear that in biosimilar 2 there is an N-terminal truncation happening that would have been very difficult to spot with the overlaying complexity of the glycans still attached. So this sort of workflow will allow you to strip all the O-glycans from a protein so that you can analyze what's left, the unmodified protein. And with that, I would like to end. I would like to end and summarize. We've talked about in-depth mapping of protein O-glycosylation using ODERATOR, both in a biopharma context and a glycoproteomics context. We talked about quantitative comparison of O-glycosylation patterns in biosimilars and originators, and also routine analysis of O-glycosylation. And then finally, the complete removal of N and O-glycans from proteins for intact mass analysis. If you would like to know more, our homepage, you will find very many posters and application notes at genovis.com. You can also contact our support team at support@genovis.com. Or if you would like to order our smart enzymes, the ones we've talked about today, or all the other ones, there's a new web shop available, or you can mail at order@genovis.com. And I understand we have a few questions.
Yeah, thanks very much, Andreas. So we have two questions here. Do these enzymes require denatured material to be active, or are they active on the native fold of the target protein?
O-glycans, due to the fact of how they are synthesized in the cell, are often accessible. So in most cases, these enzymes will have access to the O-glycans on the native fold. You don't need to denature. All of the data I've shown today is done on non-denatured materials in the native folded state. However, if there is an issue or if denaturation is required for any other case, the enzymes will also work on denatured material, absolutely.
Okay, that's good. Great. And the second question then is, do the sialic acids need to be removed for the activity of ODERATOR?
This is a bit of a tricky assay there. So ODERATOR will digest sialylated material, sialylated O-glycans, but at a very much reduced rate. So the detail, the analytical depth you will achieve is much lower when you leave the sialic acid intact. That's why we recommend you use Sialexo and remove the sialic acids. However, if you want to get information on sialylation, there's a possibility to do this with just operated digestion, no Sialexo. And you will get some site-specific structural information. We have shown some data on this on the poster we presented at last year's ASMS. It can be found on our website.
All right. Thank you.
Yes, and with that, we'd like to end. Thank you very much for watching, and until next time.